Welcome to our lab podcast on the urinary system. We start off with a fairly low magnification slide that's basically going to view a section through the kidney. And in this particular section, we start with the outside. This is where we're going to have the capsular surface of the kidney. And as we move further down, this is going to be the cortex of the kidney. And we can recognize the cortex by these little spherical objects that we see in the field. These are going to be our renal corpuscles. And you can see these scattered throughout. And as we get approximately in this location, especially where we see some of these larger cross sections of vessels, this is going to be the juxtamedullary border between the cortex and the medulla. So our medulla will be here. Here we have our cortex. Now, as we look in this view, you can see that many of the renal corpuscles are going to be aligned in this column. And you see we have a space where we don't have renal corpuscles. And then once again, we have corpuscles, space, corpuscles. These are going to be our cortical columns. And these, which are basically going to be our collecting ducts streaming down from our nephrons toward the medulla. This is going to be our medullary ray that, it appears to be projecting up into the cortex, but we call these the medullary rays. A little bit higher magnification, still looking in our cortical region. Here we see 3 of our renal corpuscles. They're going to be surrounded by our cortical labyrinths, which is going to include our proximal and distal convoluted tubules. And then here we have more in longitudinal section. These are going to be our collecting ducts in our medullary ray. And then we get over to the other side. Here we have another renal corpuscle, cortical labyrinths, and then the next ray would be out of this plane of section. Now, looking more closely at our renal corpuscle, what we're going to see here, and recall, a renal corpuscle is going to be essentially our glomerulus surrounded by Bowman's capsule. And in this particular instance, what we're seeing in the inset is going to be our juxtaglomerular apparatus. Now, in this case, we have a lot to look at, and we're going to talk about several of these things. First, we are going to have certainly our glomerulus that we can see in our field here. These are our capillary loops. Now, mixed within this glomerulus, we're going to have some intraglomerular mesangial cells. We can't really pick them out adjacent from our endothelial cells of the capillary, so we're not going to do that in lab. But on the very outside, when you see nuclei on the very outside of this particular structure, that's going to be our visceral layer of Bowman's capsule. Those are going to be our podocytes. Here we can see one as well. Now, these epithelial cells lining this outer portion, this is going to be the parietal layer of Bowman's capsule, and this is going to be Bowman's space. Now, at this point, what we can see, we have an arteriole coming in, and we can see our very flattened endothelial cells of our arteriole. We're going to have another arteriole here in this case. And then you see these really red staining cells that are adjacent to the lumen of the arteriole. These are going to be our JG cells, our juxtaglomerular cells. Now, these are the ones that are going to be detecting our pressure differences that we have here in the arteriole. We can clearly see all of these nuclei lined up very, very closely together. These are going to be our macula densa cells, and we'll probably look at these a little more closely as we go through this as well. Now, remember, the macula densa is part of our distal convoluted tubule. So as we follow this back, we can see a number of nuclei present in our distal convoluted tubule. We see this is fairly short in terms of the height of the cuboidal cells, all indication that we're looking at distal convoluted tubule. We have no number of sections of those in here as well. Here's a section of distal convoluted tubule. And once again, being able to see a lumen is also indicative of that as well. Here we see a distal convoluted tubule. We can see the lumen, but the number of nuclei are going to point to that as well. Now, in this particular section, you can see the nuclei really have these dark spots in the middle of them, and you don't see as many nuclei. You can't really distinguish the borders of these cells, and the lumen is somewhat occluded. This is going to be a proximal convoluted tubule. Here we see more of the proximal convoluted tubule. It goes out of the plane of section. More proximal convoluted tubules, but all the rest of these are going to be sections through distal convoluted tubule that we're looking at here. Now again, a little bit higher magnification, looking at our juxtaglomerular apparatus. Again, macula densa, portion of the distal convoluted tubule where it comes back beside our vascular pole of the renal corpuscle. And here we have our juxtaglomerular cells, and these are going to be our extraglomerular mesangial cells sort of spread out here in the middle. Now, pulling back away, we're looking more at our cortical labyrinth now, and by and large, the majority of tubules you're going to see are going to be proximal convoluted tubule as we look in the cortex. So here we have our renal corpuscle, Bowman's space, parietal layer, and again, as we're looking at these cross sections, the ones that are taller, the ones that have fewer nuclei present, these are going to be our proximal convoluted tubule. The ones where we can clearly see nuclei, many nuclei, we can see a distinct lumen, those are going to be our distal convoluted tubule. And you can compare and contrast these in a number of places. Here's a distal, there's a proximal tubule. Now you're also going to see structures such as this, and this is going to be part of our peritubular capillary network. A lot of capillaries that are going to be around in and among all of our tubules, supplying them with the oxygen and nutrients that they need. Now, here's another view of our renal corpuscle. Once again, we start on the outside. We can see our parietal layer of Bowman's capsule, Bowman's space. We have our loops of our capillaries. This is going to be, we can't really see our afferent and efferent arterioles, but we certainly don't see our proximal tubule exiting. So we really can't see any of our poles, but this is probably going to be up near the vascular pole. Again, on the outside, we're going to see the cells that are our podocytes. And then we can see our capillary loops on the inside. And situated between, we are going to have our mesangial cells. Up in here, these would be extraglomerular mesangial cells. And in the middle, down in here, these are going to be our intraglomerular mesangial cells. Here we see a couple of more renal corpuscles. We're not going to spend a lot of time here. This is not one of my favorite views simply because the lumen of these proximal tubules is just exploded. Okay, and these don't look really as tall, but look how few nuclei you can see present here versus almost every nuclei you can see here. This is a distal tubule, and the rest of these are proximal convoluted tubules. But what we're looking at in this particular renal corpuscle, you can see the parietal layer, you can see the glomerulus, but notice how we go right into a proximal convoluted tubule. That means that this area we're looking at, this is going to be the urinary pole for this renal corpuscle. Now as we start to look at our cortical medullary junction, we're starting to move away from that. Now we're going to be looking at our collecting ducts and collecting tubules. So remember, this is going to be part of our medullary ray as we're extending down into the medulla. And here we see some fairly tall cuboidal cells. And as we look at these tall cuboidal cells, very open diameter lumen, this is going to be our first indication that we're looking at a collecting duct. Now, As we look even further, we can see some of these taller, more reddish looking cells, dome-shaped on top, and you can see these nuclei are even in a different plane. These are going to be our intercalated cells or our PEG cells, and that's also going to help us know we're looking at a collecting duct that's present here. Now, as we look at our other tubules, we're going to get an idea of their relationship. You notice we have tissue and cells that look very much like a proximal tubule, and here we can see a lot of nuclei, shorter cells. This looks very much like a distal convoluted tubule. So we got a collecting duct. So if we're in the medulla, this is going to be part of our loop of Henle. This is going to be our thick ascending limb, and this is going to be our thick descending limb because of their similarities to what we saw with distal convoluted tubule and proximal convoluted tubule. Here we are in the medulla once again, and what we can recognize in this right away is we have a very, very thin set of cells here, nuclei in every place, almost squamous in nature. This is going to be a thin limb of our loop of Henle. We're only going to find this really down in our medulla. And as we look at this, we can see structures such as this somewhat shares characteristics. This looks similar in some ways to our proximal convoluted tubule in a number of nuclei, but you see how much shorter we have in the height of our cells. So this is going to be one of our thick limbs, and this is going to kind of be a transition. I'm sort of leaning a little bit because of the shortness of these, leaning a little bit toward thick ascending limb, but this is not one that I would ask you on our practical exam. This is sort of one of those gray areas in between. Once again, we have a nice view of a collecting duct. We're also going to see some of the thick portions of our loop of Henle, and we're not going to worry about those quite so much. I'm much more interested in you being able to identify collecting ducts and distinguishing them and the cells of the collecting ducts versus what we'll find in our cortical labyrinth between proximal and distal. So we're going to focus on this collecting duct. Again, mostly what we're looking at are principal cells, but then again, we will see our red staining cells, cells projecting and dome-shaped at the surface. These are going to be our intercalated or PEG cells that are characteristic of our collecting ducts. Now, continuing in our look through the medulla, what we're going to be looking at here is we're looking at a sort of a weird section through a collecting duct, and that's what this tangential material here is going to be. We don't see the lumen of this at all. Here's one of our thick limbs, but what we're clearly going to be able to see, we've got red blood cells, and so we know we're going to be part of our peritubular capillary network or part of our vasa rectae. We don't know arterial versus vena rectae at this point, but we know we're in part of the circulatory system. But then we get to these really flattened cells close together, very little, if anything, in the way of lumen, but no red blood cells whatsoever. This is part of our thin limb of the loop of Henle. So this is pretty easy to distinguish because of the absence of our red blood cells. And you see how tall these nuclei are? They're not nearly as flattened and tapered as our endothelial cells in our blood vessels. So that's another indication we're looking at portions of our thin limb of the loop of Henle. Now, as we look at this image, all of these longitudinal sections, we are looking at our collecting ducts as they're moving into our larger diameter papillary ducts, and we can actually see a papillae. We're getting into our area cribrosa here at the very end. So much of what we're looking at here are going to be our collecting ducts streaming in, and you see how they coalesce together into these very, very large ducts of Bellini or papillary ducts, and they coalesce together until they empty into this space that's occupied as one of our calyces. Now, this image is going to be in that juxtamedullary boundary region. We know that because we've got a renal corpuscle here. We can see some more renal corpuscles, and then we get into sections through mostly our collecting ducts and tubules and loops of Henle. But what we're looking at in this juxtamedullary boundary is essentially going to be sections through part of our vasculature. We have two arteries here, sections through two arteries, and then we have a section through a vein. And at this junction, the juxtamedullary boundary, these are going to be arcuate vessels that are running along, following this juxtamedullary border along this curvature or this arch, hence the name arcuate arteries and arcuate veins. Now, we've stained things a little differently, pulled back, looking at a nice overview of our kidney, and we can, especially in this, see our renal pyramids that are coming down here. We've got our cortex on the outside. We got our medulla. Here we can see a renal papillae and one of our first calyces that we're going to have present here. And remember, on these calyces, that's where we're going to find our transitional epithelium. Our arcuate vessels, like we just saw, are going to be running right along this border that we see right in here. And these folds, I mean, it's kind of a weird cut. I'm not exactly sure because we got cortex tissue sort of curving around here. So this might be a tangential cut through maybe what they're referring to as a kidney lobule. But once again, we see our renal pyramids, the papillae, and here we see our calyces. Now we're back to our low magnification view, going a little bit more detail as we begin to look at structures. And here we're going to look at our vessels. Once again, we have our arcuate vessels that are going to be following this curvature along, and most of these cross sections is what we're going to see. And then the vessels that are running up through our medullary rays and through our cortical columns, these are going to be our interlobular. See, we have the lobars, interlobar down here. These are interlobular vessels that are running up through the cortex, perpendicular to these arcuate vessels. Another view of cortex. Here we have the capsule of the kidney at the surface. And can you identify these structures that we see in the field? All of these are going to be proximal convoluted tubule. We may see, this may be distal, but again, it's very difficult to tell since we don't see the majority of that there. So we're looking at proximal convoluted tubule sections, and you can actually see the striations here along these cells. But notice, you don't see every nuclei. The nuclei that you do see have this nice dark nucleolus, and we don't see the lateral cell boundaries. Lumen is occluded. Very characteristic of what we see with proximal convoluted tubule. Once again, we are looking in the cortex. We can see our renal corpuscles quite nicely. We can see our interlobular artery, possibly interlobular vein. Can't really tell the thickness of that wall. There's not much thickness there. And this is pretty nice because we can see, even in this magnification, we can see our proximal convoluted tubules, and then in these sections, these darker nuclei all lined up together, distal convoluted tubule. And then once we get into our medullary ray, we're going to be looking at collecting tubules. Here's a nice view looking at a renal corpuscle, basically the parietal layer of Bowman's capsule. Out on the outside, we're going to have our visceral layer, our podocytes. And here you can see red blood cells present in the capillary loops of the glomerulus. Now, looking at that relationship between our podocytes and the endothelial cells, here we're shifting to electron microscopy. On the inside, this is our capillary loop. On the inside, we're going to have our fenestrated type epithelium, the endothelial capillaries here. We got our basement membrane. Remember, 2 lamina raras with a lamina densa between. And here we can see the pedicels or secondary processes of our podocytes with their filtration slits and filtration membranes between. Here you can see a secondary process or a pedicel actually touching down, and this is a primary process of a podocyte, and here's the nucleus of that associated podocyte, and you can see more of those pedicels attaching downward. Higher magnification, again, we've got our fenestrated endothelium. We can see our pedicels, and there's that nice view of the pedicel coming down. There you can see the slit between the pedicels, and there that little line between, that is going to be our filtration membrane that's present within the slits. The light areas we see here are lamina rara interna and externa, and then lamina densa, the dark material, lining right down the middle. Another view of the same endothelium at the top, basement membrane, pedicels, and you can somewhat see the membranes possibly there and there. You don't necessarily see them in each one. But those are going to be our 3 filtration mechanisms that allow plasma to move out into Bowman's space and be our glomerular ultrafiltrate. Now, as we leave the kidney at the renal pelvis, the ureter, this is going to pick up that material. And here we can see our folded membrane. This is our mucosa. We've got our submucosa connective tissue, and then our muscularis on the outside. And then outside of that, we have adventitia. Look a little bit more closely. Here we can see our mucosa. This is going to be transitional epithelium. It's fairly flattened at this point. There we might have a binucleated cell, but this is transitional epithelium, submucosa, muscularis, adventitia out here. Here, looking more closely, we can see the fairly large, here's a nice dome-shaped cell, part of our transitional epithelium. Looking along, see if we can see any binucleated cells. Again, possibly a binucleated cell there. But these very large cells on the surface, that's going to be typical of what we'll see with our transitional epithelium. Not very much to talk about here, just our typical smooth muscle layers. We see our circular and longitudinal muscle on the inside and outside respectively. It's part of the ureter still. And then another more close-up look at our rounded dome-shaped transitional epithelium. Remember, it's going to look fairly pseudostratified, if not stratified, but when you look at that surface and see those dome-shaped cells, even if you don't see the binucleated cells, that's your tip-off that you're looking at transitional epithelium. Another view there, we can see our, in the very center at the top, our binucleated cells, characteristic of transitional. And then we can see our submucosa connective tissue. Now, we don't have additional slides, but again, recall that the bladder will also have our transitional epithelium, much larger luminal diameter, however. And then our urethra, depending on where we are, if we are closer to the bladder itself, we're going to have some transitional, but that's going to become our perikeratinized mucosa as we get closer and closer to the urethral orifice and to the outside of the body in both males and females. In males, we will have the prostatic urethra. So if you see glandular tissue completely surrounding a lumen, then you know it's urethra and it's prostatic urethra. If you see the urethra within the spongy caverosum in the penis, once again, that's an easy tip-off that you're looking at the urethra.